we're going to talk about the male reproductive physiology. Okay, so first of all, we should just appreciate the anatomy, that the site of sperm production, which is the responsibility of the male reproductive system, occurs in the testes. Okay, so the testes are enclosed in the scrotum. The testes, specifically the seminiferous tubule of the testes, are responsible for sperm production, right? Okay, so the testes will transport their contents into the ductal system. Okay, here we can see the epididymis. It connects to the testes, to the posterior wall of the testes. Then from there it goes to the vas deferens, which exits the scrotal sac, okay, through the spermatic cord. Then from there it goes to the posterior surface of the bladder, then joins with an accessory gland of the male reproductive system called the seminal vesicle to form the ejaculatory duct. Okay, so here we're just talking about the pathway which is taken by the what? By the sperm. Then from there it enters into the prostate gland. Okay, then from there it enters into the urethra. Then afterwards they are ejaculated out, okay, during the male sexual act. So that's like the anatomy in a nutshell. Okay, let's go a little bit more in detail. So initially these testes are found in the abdominal region in uterine life. Okay, then at birth they descend into the scrotal sac. Okay, if they fail to descend into the scrotal sac, we call that condition cryptorchidism. Okay, the testes, they perform optimally for the process of sperm production in the scrotal sac, which has a temperature which is 1 to 2 degrees lower than the temperature in the what? In the body. Okay, so the scrotum has musculature. It has the dartos muscle, which just causes the wrinkliness of the scrotum, and the cremasteric muscle, which pulls the testes up or down depending upon the temperature of the day. Okay, to help to regulate optimum temperature for the testes. So this is like a more nicer view of the testes. Okay, so the testes is composed of a fibrous capsule, okay, which you call the tunica albuginea, right? So the tunica albuginea will separate these seminiferous tubules, which are coiled tubules, which are the site of sperm production. They will divide them into lobes. Okay, so the sperms are produced via a process which you call spermatogenesis, where a gamete, a sex gamete, okay, the male sex gamete divides. It divides via mitosis first, then meiosis to form spermatids. Then the spermatids then form spermatozoa. Okay, we'll talk about that later. So the newly formed sperm, okay, so initially their sperm production occurs at the base of the seminiferous tubule. Then they move towards the lumen. Then they are transported from the seminiferous tubules via the straight tubules into this medium sternum of the testes, which you call the reti testes. Okay, and let's take note that at this point the sperm are not motile. Although they will have a tail, they are not motile. So the fluids in here will help to propel the sperm into the reti testes. Okay, then from there we have like some cilia in the efferent duct too, which will help like in the in the sweeping action of the testes into the epididymis. Okay. So the epididymis is this structure here. It's a muscular tube, right, which is about 6 meters, 7 meters there. Okay, which has a head, body, and tail. It transports the sperm, right? Since we say that it's still immortal, it's also the site of storage, temporary storage, and maturation of the sperm. So these muscular tubes, they will contract in a peristaltic motion, which will help in the transportation of the sperm. They also have stereocilia, which will help in the propulsion of sperm. Then from there they enter into the vas deferens. Okay, here in the posterior aspect of the testes, the vas deferens. Then the vas deferens also is a muscular tube, okay, which connects the testes, or which then transports the sperm into the urethra, okay, via connecting with the accessory gland which I showed you, which is the seminal vesicle. Okay, done. So this is the process of spermatogenesis which we're talking about. It starts in males at puberty. Okay, so the male individual is said to be at puberty maybe around 12 to 14 years after birth. Okay, so a primordial germ cell initially will first of all enter into the testes. Okay, from there it's now called the spermatogonium. Okay, spermatogonium singular, and this cell will be diploid, and it will have the ability to divide by mitosis. Okay, so mitosis is a division which preserves the chromosome number. So you have the same number of chromosomes as the parent cell. Okay, so initially the chromosomes which contain the genetic material will be single-stranded. Okay, so they will divide by mitosis and form daughter cells. One will remain behind, which we call like let me do this. We call this one the spermatogonium A. Okay, it will remain in the seminiferous tubule for later use. Then the B1 will further divide and move closer to the luminal compartment. Okay, so we say that this process happens at the base of the seminiferous tubule. So the spermatogonia B would further divide and move closer to the lumen so that it can be transported out of the seminiferous tubule. Okay, so this one will further divide via mitosis to form what are called primary spermatocytes. So these primary spermatocytes, as you can see, are double stranded. Okay, unlike these single stranded spermatogonia. Okay, so these primary spermatocytes then undergo a division which we call meiosis, in which their chromosomes become half the number as the parent cell, as you can see here. The first meiotic division will produce secondary spermatocytes which have a double-stranded nucleus, as you can see here. Okay, but they have half the number of chromosomes as the parent cell. Then the second meiotic division will produce what are called spermatids, which are now single-stranded. Okay, and they contain half the number of chromosomes as the parent cell. Then the last stage is spermiogenesis, in which these sperm, they mature to form spermatozoa. In this process, they will lose some of the cytoplasmic contents, and the nucleus condenses. They develop a tail, and they develop a acrosomal cap. Let me show you. Okay, so this is like the spermatozoa, which is a head, a midpiece, and a tail. Okay, so what happens is during spermiogenesis is the Golgi bodies. They will condense to form the acrosomal cap, which will contain hydrolytic enzymes which will help to degrade the cap or the coat of the oocyte, right? The nucleus will condense. The nucleus contains the genetic material. Then the mitochondria will form like this nice structure, and this nice structure we call this the mid piece. They will be part of the mid piece, and the mitochondria will be like in this spindle, right? They will be like in this spindle here, right, in which they are arranged properly to allow they will be like the producers of ATP, which is like the energy currents of the sperm, which will cause like the beating action of the tail of the sperm. And the tail of the sperm is made up of microtubules, and like we call this part the flagellum of the sperm. Okay. And now, since the sperm has lost most of this contents, it would depend now upon external sources for its nutrition. And these external sources are the fluids which are secreted by the accessory glands. Okay. Now, going back to the seminiferous tubule. In the seminiferous tubule, we have cells which you call Sertoli cells, which also called nest cells or sustentacular cells. These cells they help to act like the support of the developing spermatids, or let's call them spermatocytes. Okay, so here we're just showing us the process of spermatogenesis. Okay, it happens at the base, then moves high up to the luminal side. Okay. So these Sertoli cells, they also form a barrier which we call the blood testis barrier to help protect these developing sperma, like to help protect the spermatozoa here. Okay, they also have secrete a androgen binding protein which binds testosterone. Testosterone is produced by the Leydig cells, which will be found in the connective tissue stroma here. So it's a lipid hormone, testosterone. We'll talk about it later. So its concentration is helped by the Sertoli cells, which bind it and provide it for these developing spermatocytes. Okay, they also secrete an anti-Mullerian hormone which help to degrade the female reproductive ductal system. Because initially during uterine life, we'll talk about it, the cells will be like, let's not say the cells, the developing fetus will be bipotential, or developing the developing oocyte, the developing child, you can say the word, or the developing fetus. Okay, let's say the developing fetus will be bipotential. To have the ability either to divide into a male or a female. So this anti-Mullerian hormone will help to degrade the female part, okay, and promote the development of the male secondary or the testes. Okay, it will degrade the development of the uterus, the fallopian tubes, and the likes. Okay, it also secretes a hormone which called inhibin, which will inhibit the pituitary secretion of FSH. So FSH is this one. It will it stimulates Sertoli cells so that they function properly. Okay, So if like we see that there is overproduction of sperms, we inhibit the source of FSH, which is from the pituitary gland. Okay. And also these tight junctions, they help to form like or divide the seminiferous tubules into a basal compartment and a luminal compartment. Then the Leydig cells, we say that they are the producers of testosterone. They are found in the what? In the connective tissue surrounding the seminiferous tubule. Okay, we'll talk more about testosterone later. So we have semen, okay, which is ejaculated during the male sexual act. We'll talk more about this later. It will contain fluids and the sperm. Okay, so the fluid part, it will have secretions from the accessory glands. Okay, so from accessory glands and also from the vas deferens. 10% from the vas deferens, 60% from the seminal vesicles, 30% from the prostate gland, and a small amount of mucus from the bulbourethral gland. Okay, so these fluids, they will help the sperm to survive the acidic environment of the vagina, and also to survive the mucus secretions from the from the cervix. Okay, because the sperm, they become fully motile in a pH of about 6 to 6.5. Okay, so these are the accessory glands, the seminal vesicles, which are found on the posterior surface of the what? Blood. Okay, they secrete mucoidal material, which contain fructose, citric acid, prostaglandins, and fibrinogen, which help protect the sperm as they are moving into the female reproductive tract. Okay, the prostaglandins will react to the cervical mucus, which will help in the sperm movement towards the what? The oviduct. Okay, this prostaglandin will also help cause the sperm to move into the fallopian tubes or oviducts, so that they can fertilize the what? The oocyte. Okay, prostaglandins, they secrete, is also an accessory gland which secretes milky fluid which will contain calcium. Calcium will help in the capacitation, okay, or yeah, the further, because the sperm cannot fertilize the oocyte if it's not capacitated. So calcium will help in making sure that there will be beating action of the tail of the what? Of the sperm. Okay, then clotting enzymes will help during sexual intercourse. It will help to make sure that the sperm, they remain in the female reproductive tract, so that they are not spewed out. Okay, so that they stick in there. Then the fibrinolysin will make sure that after they've stuck for a while, they can now move to fertilize the what? The oocyte. They also secrete alkaline solutions which will neutralize the acidic environment of the what? The vagina. They also secrete a fluid which is called the prostate specific antigen, which breaks down semen and helps to release the sperm. Okay. Since we say that first of all we form like a clot, then the clot must be washed so that the sperm might be released from the semen and go to fertilize the what? The oocyte. So yeah, this is the female reproductive tract, which has a vagina, the cervix, the uterus, and the fallopian tubes. Fertilization happens in the fallopian tubes. Okay, somewhere here. So the sperm have to move and move. So this movement requires that the sperm should be capacitated, and capacitation is aided by the calcium ions, and also by they're aided by the calcium ions, so that they can have that beating action. And also the environment of the vagina also facilitates capacitation. We'll talk about that later. Okay, So sperm count and motility. So we need the sperm to have a normal shape like the one I showed you before, so that they can be able to fertilize the oocyte. Okay, and we also need the sperms to be sufficient in quantity for them to reach the oocyte. Okay, so usually during the sexual act, in a 3.5 ml solution of semen, it contains about 120 million sperms. So yeah, this is like the average estimate. When the number during the sexual act is 20 million, we say that it is termed oligospermia. So we are saying the sperm count is low. Then if during the sexual act there is absence of sperm, we call that azoospermia. Something to appreciate. And we can even have these sperm which have defective shapes, double-headed, coiled. Yeah, so we should just appreciate the fact that we can have absence of sperm, we can have insufficient sperm being during sexual act, and we can also have over excess of sperm during the sexual act.